Okay, it's going.
Okay. This is the Tong et al. Paper overview. It's about the synaptic release of GABA by AGRP neurons is required for normal regulation of energy balance by Rachel Grazda and Jake Keski.
So the main problem this research examines is the importance of GABA neurotransmitters in the hypothalamus, and specifically in terms of the AGRP neuron.
GABA is required for energy expenditure and food intake, and for ghrelin stimulated food intake, which has an effect on body weight. Ghrelin is the hunger hormone and it increases food intake. The AGRP neuron is a major target of ghrelin. Um, it releases GABA, AGRP, and the neuropeptide Y. That's three things. And neuropeptide is produced in the brain, and it's one of the most potent.
So while the AGRP neuron releases these three components, prior research has noted that when you delete the genes for the AGRP and the MPY, that it has little effect on the body weight and energy expenditure, which is the main function of the neuron. So that means that GABA is obviously the important factor here.
Okay, so VGAT is required for. Oh wait, yeah, VGAT is required for GABA release and pre and presynaptic release. In order to further test GABA, they disrupt the VGAT pathway so GABA cannot be released. Yeah, so they cut the VGAT pathway.
In order to disrupt the VGAT pathway and test what GABA does, they used Kralox recombination and created two types of mice and then bred them. Kralox recombination is a site-specific recombinase technology used to carry out deletions, insertions, stuff like that. In this case, they are deleting exon 2 in the wild type gene of VGAT up here. And to target this exon for deletion, they used a technique called floxing, which is just sandwiching the exon 2 between LOX P. Then the Cray enzyme can come in, recognize that flox sequence, and act like scissors to cut it out. But in order for Cray to be expressed in these AGRP neurons, they need to create this second type of mice, which is the bottom here, AGRP iris Cray mice. And these mice just have a site-specific promoter that allows Cray to be expressed in the AGRP neurons, and only those AGRP neurons. So then they just bred the VGAT flox mice with the AGRP iris Cray mice, and the offspring have not only the flox site, but also they have Cray expressed so that VGAT is disrupted and GABA cannot be released from the cell.
So they measured GABA and mediated currents in the postsynaptic cell, and in the control group, 7 out of 13 of the mice had GABA, and in the experimental group, 0 out of 12 had GABA. The deletion of VGAT prevents GABA from releasing in the AGRP neurons.
So now that we have confirmed GABA release was disrupted, we can take a look at the results they found. So they already know generally that the normal function of the AGRP neuron and therefore GABA is to restrain energy expenditure. So that means the mice they created have no restraint on energy expenditure. So energy expenditure just equals basal metabolism plus digestion and physical activity. So they saw an increase in locomotor activity. They saw an increase in O2 consumption because they are expending more energy, and they saw a reduction in respiratory exchange ratio since oxygen is the bottom of that ratio, and that is increasing. And prior research has shown that a lower respiratory exchange ratio indicates that fat is the primary fuel source, and if you're breaking down fat, then you're going to have a lower fat accumulation and you're going to have a lower body weight. So these are just the graphs they had in the paper. The left two graphs indicate the decreased body weight here on the bottom here, and then the second two graphs indicate that they had a higher O2 consumption.
Okay, so AGRP neurons are a major target of ghrelin, the hunger hormone, which stimulates appetite, increases food intake, and promotes fat storage. Mice with defective VGAT, lean mice, had decreased response to ghrelin, This indicates that GABA release from AGRP is an important mediator of ghrelin's stimulatory effect on food intake. GABA is required for energy expenditure, ghrelin stimulated food intake, and they found this out by deleting VGAT. It could be the release of GABA mediated inhibition of POMC neurons, which are neurons in the hypothalamus that play a role in metabolism, which have large stimulatory effect on thermogenesis.
So in terms of its importance, I could totally see people using the technique of disrupting VGAT and creating some sort of treatment for people who are prone to obesity and need to lose weight. But I also could see this becoming a bit dangerous if it becomes mainstream or over the counter because people think that they could overeat, eat unhealthy, whatever, and they're not going to gain weight. They're going to stay lean. But obviously, you're just shoving a bunch of crappy nutrients into your system and you're not giving your body the proper ones, which could lead to a lot of issues. So, yeah, it would have to be highly regulated.